Welcome back. In this lecture, I continue a discussion of the science behind cancer treatment. Previous two lectures, I've discussed surgery and radiation. In this lecture, I turn to chemotherapy, the use of drugs to treat cancer. My story begins with World War II. The attack was unexpected. That afternoon, a local commander had said that things were so secure in the port of Bari, Italy, that he would regard it as a personal affront if the enemy air force attacked. But in December 1943, attack they did. At about 7:30 in the evening, over 100 German bombers sank 17 Allied ships in about 20 minutes. Hundreds of sailors were killed. Now, the ships were there to supply Allied armies pushing north into Italy. One of the U.S. ships damaged was called the John Harvey. And the John Harvey was carrying a secret cargo. This cargo mixed with oil in the surrounding water, and the sailors who jumped out were coated with it. The next day, the sailors complained of blindness and burns. In addition, some gases were released in the explosions of the ships that you can see in the photograph. And these gases drifted on shore. There was a garlicky odor to them. Hundreds of civilians in Bari, Italy, came to our U.S. Navy medics the next day with the same symptoms. What had caused the symptoms? The U.S. Surgeon General ordered an investigation and found, much to the surprise, that they were caused by mustard gas. Mustard gas was supposedly illegal. In World War I, mustard gas had been used by the German army. It causes blisters, chemical burns in the eyes, and blindness. Tens of thousands of soldiers were incapacitated and killed. In 1925, the major nations of the world signed the Geneva Protocol outlawing chemical weapons. By the way, the U.S. didn't ratify this for 50 years. These weapons were deemed too terrible to use in warfare by civilized nations. But World War II came along, and the Allies suspected that Germans were again developing mustard gas and other gas warfare elements. The U.S. therefore developed a secret stockpile of mustard gas. And on the John Harvey, there were 2,000 mustard gas bombs in this secret store. The Allied High Command at the time covered up the incident at Bari, lest the Germans be provoked. Now, the previous use of mustard gas in World War I led to some interesting observations. Soldiers exposed to mustard gas had lowered blood counts, especially in white blood cells. So alert doctors said, well, some blood cancers have too many white blood cells, and this mustard gas lowers white blood cells, so why don't we use something like mustard gas to treat people? In 1943, the Bari incident I've just described also caused the suppression of white blood cells in the U.S. seamen and in the Italians who were exposed. This stimulated this cancer treatment idea even more. Let's use this to reduce white blood cells in patients who have cancers of white blood cells, leukemias. But there were two problems in doing this. First, it was illegal. So any clinical use is going to be secret. And second, sulfur mustards, you can see on the graphic that mustard gas contains an S or sulfur atom, are very toxic. Obviously, that's why they were used in battle. So instead, scientists made a nitrogen mustard. If you can see in this graphic that I'm not asking you to be a chemist. The sulfur has been replaced with a nitrogen. This molecule, the new nitrogen-containing molecule, is called mustine. And after World War II, this research was declassified and these ideas, and an open clinical trial was done, and they got good results for treating leukemia. This was around the time, the late 1940s, early 1950s, when the role of DNA was discovered in biology. And you recall, in 1953 was the time when the famous double helix model was unveiled. When people studied how mustine works, not surprisingly, they found that mustine works on DNA. Here was something that killed cells, and as you know from the course so far, things that kill cells sometimes react with DNA. In this case, the mustine would bind to guanine or G residues on DNA. So here is a G on DNA, and the mustine molecule will bind to two Gs on opposite strands. Now, G is opposite C on the DNA, but there's going to be a G nearby on the other strand, and so it'll form kind of an oblique cross-link on DNA. And here is the mustine cross-linking in a very tight bond, DNA. So when DNA tries to unwind, and DNA tries to unwind every time it duplicates itself, the strands have to separate, or locally it'll unwind when it expresses itself, it makes this RNA copy. Whenever DNA tries to unwind, it can't. The DNA, it's all stuck. Okay, the DNA is stuck together. It can't unwind at all. And so the cell dies. It will essentially go into programmed cell death or apoptosis. To summarize, this is an early example of chemotherapy, using chemicals for therapy. The use of drugs took off really from this example as well as some others at the time. It's also a really interesting example of clinical research. When people started using mustine to treat leukemia, they didn't know how it worked inside the cell at all. All they knew is that it suppressed white blood cells and was useful for treating leukemia. So some research in medicine, and you're going to see this as I talk about chemotherapy, is empirical. That is, they're just looking to see what works and experimenting with what works, not knowing the mechanism. The mechanism comes later. Hopefully in the future, we're doing more research on finding the mechanism first, proposing the mechanism, and then designing a drug. In this and the next lecture, I'm going to describe chemotherapy. I'm going to do it in several ways. First, I'll describe the pathway taken by a drug to the tumor. And then I'll describe cancer combinations of chemotherapy. Why, when you see a person getting cancer chemotherapy, there's more than one drug typically used to treat it. Third, today, in this lecture, I'll describe how drugs are developed from nature, and I'll give you some examples of that. In the next lecture, we'll go on with some specific examples of cancer chemotherapy drugs. By no means an exhaustive discussion, but a discussion to show you some of the science behind some typical drugs. So I'll describe drugs that attack DNA, drugs that attack other cell processes, and then turn to the really nasty part of chemotherapy, which, if you're familiar with cancer at all, you know about, which is side effects and resistance to chemotherapy. And finally, deal with the most optimistic aspect of chemotherapy, which is the new targeted therapies that I described in the very first lecture of the course. You recall a targeted therapy for treating leukemia in this basketball player. You will see words describing chemotherapy in three different settings, three different situations in cancer. The first is called primary chemotherapy. This is the initial treatment for localized cancer. Typically, it's being done, cancer might be localized, and you might shrink it with chemotherapy prior to surgery. That's primary chemotherapy. The second type of chemotherapy is called induction chemotherapy, and this is treatment for advanced cancer, cancer typically metastatic, typically spread. It's the most common use of chemotherapy. The third term you will see is called adjuvant chemotherapy. And adjuvant chemotherapy is treatment after another method is used for primary. So, for example, a surgeon might remove a breast cancer and then chemotherapy is given to affect other locations for the tumor in the body. So let's look first at the pathway taken by a drug to a tumor. And as the graphic indicates, it has many steps. What I'm giving you now in the next couple of minutes is a quick tour of an entire field of science. The field is called pharmacology. Pharmacology is the study of drugs and their interactions with the human or animal body. This is opposed to pharmacy, by the way. Pharmacy is the preparation and dispensation of drugs. So pharmacology is the scientific study of how drugs work. The effectiveness of chemotherapy against cancer depends on three things. First, the biology of the tumor, of course. The tumor has to be sensitive to the chemotherapy killing the tumor. Second, the pharmacology of the agents used, and I'm going to describe that in a moment. And third, importantly, the clinical condition of the patient. Can the patient tolerate the chemotherapy side effects? For good effectiveness, typically you want to kill 99.999% of the cells. Sounds like a big number. Isn't that great? Let's kill 99.999% of the cells. But remember, a 1 centimeter tumor, that's about this big, you can see my hands here, a 1 centimeter tumor has 1 billion cells. So you kill 99.999%, you got 10,000 cells still alive. The key to the effectiveness in tumor biology, that is one of the important aspects of the effectiveness of chemotherapy, the key to tumor biology and the pharmacology interaction is the selectivity of the drug. You want the drug to kill the tumor and not the normal cells. You'll recall this graph from the previous lecture. This graph describes on the up and down axis the killing or the response of the cells, and the x-axis is the dose. So tumor response and dose. What we want is a therapeutic index that's wide, so that when you give the drug to a cancer patient, it kills the cancer cells and does nothing to normal cells. That's the ideal. Unfortunately, the real world is different. In the real world, the therapeutic window is very narrow, and we damage a lot of normal cells when we damage tumor cells. Now, tumor cells are more sensitive typically to chemotherapy than normal cells for several reasons. First of all, the DNA of tumor cells is already damaged. You'll recall, a lot of tumors begin with DNA damage and accumulate DNA damage through mutations. And a little bit more damage may tip the tumor over into apoptosis, programmed cell death. Because when there is enough DNA damage in the cell, there are molecules that scan along the DNA and essentially tell the cell, look, you are so damaged, it's time to commit suicide, programmed cell death. And I mentioned before that our concept of cells being killed in chemotherapy and in radiation and radiation therapy has changed over the last 20 years. From we're killing the cells to we're inducing the cells to kill themselves. So that's the first thing, DNA is damaged. Second, there's a high rate of cell division. Well, that's going to mean that any damage we do to DNA will be permanent. Or another way to look at it would be if it's so sensitive to cell division, it's dividing very rapidly, it might be extra sensitive to molecules that inhibit cell division process itself. Third, many tumors make new proteins. You'll recall chronic myelogenous leukemia in the very first lecture of the course, making this new fusion protein because of the shuffling of the chromosomes. That can be a target for drugs. And fourth, many tumors have poor DNA repair, either through mutation or because it's been damaged during the treatment and other treatments. And so poor DNA repair makes the tumor more sensitive to chemotherapy as well. It's already mutated. If you take a drug, and take a capsule in your of aspirin or something like this, or any drug, and this includes cancer chemotherapy, the drug has a life cycle in the body. And we can plot on this life cycle, we can do a graph. How much of the drug is in the blood plasma, because that's where it all ends up, because it's being transported all over the body. So if you take a drug and it gets into the stomach, ultimately goes into your intestines and gets into the blood system. So it's in the blood. We're looking over time. And here's time. And it can be minutes or hours. It depends on the drug. What happens is you see there's a great increase after you take the drug because it takes time for it to get into the blood and to get a maximum level. And then it disappears over time. And we call the curve, the area here, the shaded area, the area under the curve. And it's very important to know how much that is for how long, because when you give a cancer patient a drug, you want to know how long it's going to be around. What's going on? Why does a drug disappear? There are two processes going on here in pharmacology that cause the drug to disappear. First, there is pharmacokinetics, things that are going on in the blood once the drug is there. So something is happening to that drug as it goes through the blood system. Second are pharmacodynamics. What's happening to the drug when it reaches the tumor? Here's a very simplified view of pharmacokinetics. This is a drug that is being administered to a woman who's a breast cancer patient. So now we have some questions to answer. The first question is, how was the drug given? Was it given orally? If it's given orally, then we have an extra set of parameters to consider, because it gets into the stomach, it dissolves there, gets into the intestines, and then gets into the blood. And all of those parameters have numbers to them, have rates of reaction. So we have to know what that is. On the other hand, you're probably familiar, many cancer-fighting drugs are given intravenously in the clinic. So an IV will go into a vein and goes right into the blood. So now we have it in the blood. And now we have to answer some questions, and you can see them in the graphic. First, it gets into the blood, and of course, it passes into the liver. And then you ask the question, what's going on in the liver? Well, we already know this system, don't we? We were introduced to this liver system that changes molecules when I discussed cancer-causing things. Now it's changing another outside molecule, something that fights cancer. So here is a molecule fighting cancer. In this case, it's a drug called tamoxifen. See it later on. It's going in the liver, and you see that it comes out of the liver system being changed slightly. Here's the change. It's got that OH group. It's becoming more soluble. It's also, in this case, becoming activated. So the question then is, I'm going to have to answer the question. How much of it is converted? At what rate? Okay. Here's another question. The drug is in the blood. The blood goes by the kidney. Does the drug get excreted by the kidney and end up in the urine? We have to have that information as well. Another question, this drug's still in the blood. Does the drug get attached to molecules in the blood? Molecules that get into the blood very often in the bloodstream start binding to proteins or other substances in the blood. How does that happen? It's going to reduce the amount of available drug in the blood if it's bound up with some protein. All the things I've mentioned so far, all these questions that we can answer, can be described mathematically by equations. We must know this information to get to the next step, which is pharmacodynamics, which is the tumor effect. In other words, finally the drug has arrived in the blood, given all those roadblocks that can happen to get it there. And now the questions are, for example, is there a good enough blood system in the tumor to get the drug there? I described angiogenesis in an early lecture. So if the tumor has really good angiogenesis, that's great for treating the tumor. The tumor is sealing its own doom, because not only is angiogenesis delivering oxygen and nutrients and stuff to the cells, it's also going to deliver drug. But if it's got poor angiogenesis, you're not going to get the drug there very well. That's the first question. Here's another question. How does the drug enter the cells? When I described cancer cells and normal cells, I described the cell boundary, this membrane, and the membrane is composed of this fat molecules that most things don't dissolve in if they're very polar. So how does it get across that membrane? Third, what happens to the drug inside the tumor cells? There may be chemical changes in the tumor cells that alter the drug and inactivate it or activate it even more. Another question, how does a drug get to its target? Supposing the target is DNA for this drug. The drug is now crossed the membrane, it's finally inside the cell, it's got to get to the cell nucleus. The cell nucleus is a separate compartment. How does it get to, and the DNA is all covered up with proteins. So how does it get to the DNA to do its thing? All the things I've just described can be described mathematically. All of these answers to all these questions, we must know all of this as we are giving therapy to a patient. So pharmacokinetics, pharmacodynamics, pharmacogenomics, genetic differences in drug sensitivity. We've dealt with this before. Almost all of the processes I have described are genetically determined. And if you inherit a variant that breaks down the drug or activates the drug more, etc., you're going to be more sensitive or insensitive to the drug. Recall the conversion of the liver that's in the graphic now. Remember, we have long-term smokers and they don't get lung cancer, and because some of them have a mutation in the gene that codes for the liver converting enzyme here, and they don't convert a lot of the cancer-causing stuff in tobacco to the active form. Same thing for activating drugs or inactivating drugs. We have to know all of that. For example, the drug I'm showing here in the graphic is called tamoxifen. It's used to treat breast cancer. We'll deal with it later. Some people inherit genetically. They inherit genes for high rates of reaction, medium rates of reaction, or low rates of reaction. So we can classify breast cancer patients into those who have a high rate of reaction. They're going to respond to tamoxifen a lot better than the patients who have little or no conversion to the active form. So just in this one system, we can actually do a genetic DNA test to determine which gene a person has, which allele, and it personalizes the care because it may not be useful to give tamoxifen to patients who can't use it that well. For the patient, all the information that the doctors have gotten so far in our discussion, pharmacokinetics, pharmacodynamics, pharmacogenomics, can help to answer the following questions. First, what's going to be the route of administration of the drug? Will it be orally? Will it be in a paste on the skin? Will it be intravenous? Second, how often are we going to give the drug? We want to maintain a decent blood level. We don't want to kill the patient. We have to watch out for side effects, etc. So we may end up with a situation in drug therapy that's very similar to what we saw in radiation, where you give a little bit at a time. And third, how much to give of the drug, how much a dose to give to get the maximum area under the curve, that is the maximum drug that's not going to harm the patient. The way they calculate this typically is body surface area, because the body surface area is a general shorthand for blood volume, how much blood a person has, and function in the kidney. It's easy to calculate. Here's the calculation of the body surface area. How to do it, it's the height and the weight, and you divide by something, you take a square root. I've calculated my body surface area there. So when you go to a clinic and you're saying how much of a drug to give, here's an actual drug regimen for a specific drug in a clinical setting that I got. Etoposide is the name of the drug. It's given 50 milligrams per meter squared, that is per body surface area, per day. IV, intravenous, days 1 to 3. It's given 3 consecutive days. Then you repeat after 3 weeks. I mentioned before, many cancer drugs are given in combinations. In early clinical trials in chemotherapy, there was very variable. There were very few cures, very few people went into remission. I'll describe that in a moment. And the reason was tumor cells are not all alike. I've mentioned a number of times in the course there are new mutations that happen in a tumor as the tumor grows. Some tumor cells are stem cells that could cause tumors, others are not. So this leads to the idea of heterogeneity of the tumor. Different cells respond to different agents. It's often empirical which drugs to use, so you try and see what's going to happen. Sometimes, however, there's logical reasoning. I'll describe that in some examples. Different drugs treat different phases of the cell cycle, for example. So here's a bunch of drugs that we'll deal with later on in the discussion and where they affect cells in the cell reproduction cycle. So here is the part of the cell cycle where DNA is made. So certain drugs are very sensitive for DNA. Here's a drug that's very sensitive as cells are dividing. Here's a drug that's very sensitive when the DNA is getting ready for cell division. One of the early observations in chemotherapy was on a disease called Hodgkin's lymphoma. And they found very quickly that in Hodgkin's lymphoma, a single drug was nowhere near as good as combinations. So here's, for example, is a single drug 50% remission. I'll describe that in a moment specifically so we can agree on what these words mean. 2 drugs, 62%, 4 drugs, 90%. It's above 90% now. Remission means, typically measured after 5 years, mean you can't find any tumor visibly by normal techniques. It doesn't necessarily mean there are no tumor cells there. They might be hiding out. But you just can't see any can't see a tumor by normal means. Cure means that there's no tumor after 10 years. Tumor control means the tumor is still there, but it's not growing. So remission, cure, and control. Let me emphasize that in chemotherapy, there are very few cures. Repeat, very few cures in chemotherapy. Overwhelmingly, we're going for short-term remissions. Now, combination chemotherapy is widely used. So here are some of the combinations. They all have these funny names, AVVD, et cetera, et cetera. You don't need to know all of these things. There are at least 60 of them out there. And they're just tumor combinations that have been useful, drug combinations in treating tumors. So Hodgkin's lymphoma is treated by these 4 drugs, doxorubicin, bleomycin, vinblastine, dicarbazine, etc. So there's all these acronyms that the doctors have for combination chemotherapy. Many cancer drugs come from nature. Recall when I described cancer-causing chemicals in nature. Plants produced many molecules for self-defense. Remember, they can't run away, so they produce these toxins. Some of these toxins damage DNA, and they cause cancer. But wait a minute, if they damage DNA, maybe they're useful to treat cancer. So the starting point of our investigation for a lot of chemotherapy is medicinal plants that have been used for millennia by traditional cultures. Over 21,000 species of plants have been used as herbal medicines that we know of. But cancer scientists are very wary of using the herbs themselves for treatments for a couple of reasons. First, very often herbal medicine is focusing on the whole body, and Western medicine focuses on the biology of a specific disease. For example, the qi, I mentioned that before in the QI. The qi in the Chinese medicine is a body function, the life force, and we're redistributing the life force when we treat cancer by manipulating the qi using herbs. Whereas Western medicine says, how do we know we're focusing on the disease? Let's focus on the biology only of the disease. So that's the first problem. Second, for herbal medicine is usually not tested in rigorous clinical trials. It's very hard to tell what the herbs are doing if they're not done in a controlled way in clinical trials, phase 1, phase 2, phase 3, etc. Third, herbal medicines are mixtures. It's a plant extract. It's got thousands of substances, including the ones that you want. There might be interacting with each other, the active ingredients. There might be more than one in the herb. And we don't even know the content very often. If you grow a tomato plant in your front yard versus your backyard, and in the front yard you fertilize it with a lot of organic fertilizer, in the back you don't, the tomatoes that come out have a somewhat different composition, actually, because of what the environment was, right? Heredity and environment determine outcome. And so herbs have to be standardized as to where they grow. There's a very good group, by the way, in Hong Kong that is trying to do this, to standardize especially Chinese medicine herbs in terms of where they grow, what the growing season was, and chemical markers for what's in the herb. In any case, the search goes on for active ingredients from traditional herbs. Let me give you one example. The example is this one. It's the Pacific yew tree, Y-E-W. The Pacific yew tree grows in the Pacific Northwest up to Alaska. It grows about 10 to 15 meters or 30 to 45 feet tall. It's been used by traditional cultures, the bark, as a medicinal for millennia to treat lung diseases and internal injuries. So people said, well, it's been used by traditional cultures. Let's take some extracts of it and we'll give it to a bunch of cancer cells in the laboratory. And what they found was the cells didn't divide. Great. Okay, then let's see if we can get the active ingredient. They isolated the active ingredient. The active ingredient here is called paclitaxel or Taxol. And they found that Taxol would also block cell division in the laboratory dish as well. At Albert Einstein College of Medicine, Yeshiva University in New York, Dr. Susan Horowitz found that Taxol works in a very unusual way. I mentioned an important part of the cell is the cytoskeleton. It's got these microtubules in it. Taxol is very unusual in that the microtubules are like these train tracks and it bundles them together. So imagine a bunch of train tracks and you grab them all together. It's going to be hard to put trains on it, right? And the tracks are kind of frozen in place by Taxol. It's very unusual. And because this was so unusual, scientists got into the clinic as soon as they could. And of course, blocking the microtubule will block these train tracks that help the chromosomes separate during cell division. So they tried it in ovarian cancer, and this is a very famous graph in Taxol research and on ovarian cancer's research as well. So we're showing here the fact that it increased survival by 50%. It extended life by 50%. It didn't necessarily increase overall survival in the end, but it extends life enormously in patients with ovarian cancer. It's terrifically successful. Note on this graph, it's very important that you see this. It's a Kaplan-Meier curve that you're familiar with. On the y-axis is percent surviving, and the x-axis is time. And it's years here, okay? That's very important that we're extending life for years. The lesson of this, natural products are very useful for cancer treatment. So in this lecture, I've described the pathway taken by a drug to a tumor with many steps, pharmacology. I've described the origin of combination chemotherapy and why that exists. And I've also given some idea of the science behind one cancer drug that came from nature. Now that we have a general view of how cancer drugs work and where they come from, we turn to the science behind specific drugs, which is the subject of the next lecture. Thank you.